Speedy.
Professor Andrew Scott, take one.
Understood.
Hello.
Hello.
Oh, yeah.
I'm good, thank you.
Okay, this is free rendering as we discussed the other day. So, I think I'd like to start. What's that? Andrew, you do that?
That's
That's
wind.
a floor polisher.
No, that's wind. You might want to open the door. Yeah.
That's wind.
Yeah.
Just put a towel.
Cut. Okay, well,
Professor Andrew Scott, take two.
cute.
Alright, and you can
Hello.
How are you?
This is going to be enough.
Not a
A little
problem.
bit distracted. Actually, coming out the Heidelberg was interesting. There's no sign outside saying this is the Austin Tower.
No, there is no. No. That could be a bit confusing.
We were orbiting Heidelberg, Austin Tower, like a malfunctioning Sputnik.
Right.
More and more frustrating.
So, did you go around outside up the hill?
We went everywhere.
So if you
I
bike
ended
ride,
up in the side area.
if you bike up there, it's referred to as the Col de Burgundy, because it's Burgundy Street, which I've done a few times. Mainly, interestingly, not for ACRF, for Tour de Cure, because they do, long cycle rides over many days to raise funds.
I'll do that. It's like
They do.
charity fundraiser.
Correct.
Yeah.
Yeah.
Do you cycle?
Yes.
Madly? Like for miles and miles and miles?
Well, I did the last one in November, and that was 140 k's a day.
So yes.
Yes.
I respect and admire what you've done. I haven't done that since I was 23 or something.
Yes. Well, I only started after my kids got a bit older, because it was impossible before then.
How did you start in science? Why did you start in science?
So my journey in science started after I finished medical school, where I graduated in medicine. I did training in internal medicine in Sydney, which is where I grew up. And as I was thinking about what I wanted to specialise in, I was very attracted to new ways in which you could actually diagnose disease through imaging. But I was also very intrigued by the possibility that the immune system could be used to manipulate the body, but also treat cancer. And so antibodies were becoming quite interesting in the literature. But of course in Australia, none of these were possible. And so after I finished my medical training in internal medicine and in nuclear medicine, I approached a very senior professor who worked at one of the biggest cancer centres in the US, in New York, Memorial Sloan Kettering Cancer Centre, to see if he had any fellowship positions available. And that led me to New York to work at Memorial Sloan Kettering, where I was for almost 3 years. When I was in New York, I didn't realise this before I went there, but the antibody program that they were developing, and they were one of the first in the world to do this, was actually led by someone who ended up being my mentor, Professor Lloyd Old, who was one of the most gifted scientists in the immunology field that anyone had seen. And so I came to know him, and I was working in his lab group as well. And that was where I learnt what I know about how antibodies could be created against cancer cells, and also how you could take these discoveries into the clinic. And so while I was in New York, I was involved with quite a number of very early clinical trials of some of the first antibodies that were used in cancer patients, many of which are now approved and used in hundreds of thousands of patients around the world every year.
How long ago is this?
Well, over 20 years ago,
So that's 2000s.
in the 90s.
Well, think about the 80s again.
Yes.
But even so, look, what's happened
Yes.
since the 60s is incredible.
Oh, absolutely.
And you're saying and this is like the mid-90s. To me, that's yesterday.
Yes, yes,
So that's, I'm just trying to wrap my head around
yes.
how fast it's been, although some people think it's slow. Is it slow or
Yeah.
fast?
So the very first clinical trials leading to approval of antibodies in cancer patients were really in the 1990s. That was when the field really started to explode. And I was fortunate enough to be in New York working at Memorial Sloan Kettering when some of these early trials were first happening. By working in the laboratories there, I was able to gain quite a deep understanding of how you could actually generate very novel antibodies and how you could then potentially take those into clinical studies. Lloyd Old, whom I worked with, was the director of the Ludwig Institute for Cancer Research, which is a global organisation. And there was a branch here in Melbourne. And one day I got a phone call from Dr. Old asking me to meet the director of the Melbourne branch of the Ludwig Institute, Tony Burgess, who happened to be in New York. And so I went and caught up with Tony, and he offered me a job, completely out of the blue, to me anyway. And that was how eventually I ended up coming to Melbourne to join the Melbourne branch of the Ludwig Institute for Cancer Research, and also because I was working in PET, positron emission tomography, which was just starting in Australia. The Austin Hospital was performing the first PET scans in the country. And so I came here to do both clinical work in advanced diagnosis of cancer and also to work on antibody-based therapeutics, which were just starting to evolve for cancer treatment.
Look, I'm just inquisitive. So where'd you grow up in Sydney?
Where?
Yeah.
So I grew up in Bexley, southern suburbs. My mother owned Scott's Pharmacy, Bexley, and was the chief pharmacist there for 50 years until she retired. And so I grew up in Bexley. I went to Sydney Technical High School. Then I went to Sydney University, and then worked in a number of hospitals in Sydney as part of my training and undergraduate time. So Royal Prince Alfred, Westmead Hospital, Royal North Shore Hospital, also Lidcombe Hospital as well.
I was looking for a catalyst, but your mother and the pharmacy won't be enough. Is that the catalyst?
Well, my mother was someone who always tried to encourage myself and my two brothers to aim high. And I became interested in medicine as a potential career, mainly from going to see the local GP on occasions. And I knew that it was a challenging area, and I thought I'd like to do something challenging. Helping people seemed to be a worthwhile profession, and so that's why I went into medicine in the first place.
What did the GP do that clearly inspired you? Sorry? Yeah, thank you. What did the GP do to you?
So it was more just observing how he interacted with patients, and because my mother, as a pharmacist, also engaged with patients that would come in for prescriptions, it just piqued my interest. I even worked as a delivery boy for her, and so I would actually ride my bike and deliver the prescriptions to people in their homes for quite a few years actually. And so it appealed to me that the challenge of trying to do medicine, and also the opportunity to engage with people and try and help them. And I think that my career in medicine, as well as in science, has been very focused on trying to help people throughout my career.
So, you got your PhD, you did your medical degree,
Yes,
and then New York.
yes.
That was Manhattan or New York State?
No, I lived in Manhattan on 63rd and York for almost 3 years. So it was about 5 blocks from Memorial Sloan Kettering on the Upper East Side. Didn't own a car, had quite a few kids. My eldest son was actually born in New York as well. So it was an interesting and very busy time.
How'd you find New York
It was very different.
after Bexley?
Yes, very different. There's the intensity of the activity in New York. You know, it's really 24 hours a day, 7 days a week. Also, living overseas, I think, is a very different experience. You get to meet so many different people, and to work at one of the major international cancer centres like Memorial Sloan Kettering, is also very different because a lot of people from around the world go there to train or to learn. And so as a fellow working there in the labs and also in the clinical areas, I met people who came from Europe, from obviously the US, Canada, from Latin America, from Asia, from around the world. Many of them became very good friends, and I'm still interacting with them now. And they've all risen to become very senior clinicians or scientists in their own institutions and back in their own countries as well. So everything was new, everything was different. The way medicine was practiced in the US was very different to here in Australia.
But tell me how. Just in general, not overly specific.
Yes. So this may or may not get into the film. So the practice of medicine in the US, when I was there and still is very specialised. And so after medical school, people often go into specialisation very quickly. Whereas here in Australia, we would do at least 4 years, sometimes longer, of rotating through many different medical areas to gain knowledge and experience. So in the US, you could specialise almost out of medical school and within 4 years be almost fully qualified in the specialty that you practice in. Whereas in Australia, it takes you 10 to 12 years to gain the same level of credentialing to be able to practice. So they were all very focused in a particular area. So as a young fellow, I would admit patients onto the ward for a clinical trial. And if they had diabetes, I would look after their insulin dosing. If they had problems with their heart, I could modify their medications. What they were used to was getting a consultation for every single thing that was wrong with the patient. So I think that there was almost a preference for having fellows from overseas to come and work in the clinical areas because to them we were extremely well qualified, but actually prepared to work as a fellow. But the laboratories were very interesting too because you had world-class people in so many different areas, all within 1 or 2 buildings. And the ability to interact with them and to hear lectures from people that were just doing outstanding work was very different from what you could experience here in Australia. And so that was why I stayed for the amount of time that I did, while also looking for an opportunity to come back to Australia as well.
So that was the Ludwig seduction that brought you back to Norway.
I would term it as a fairly unique opportunity for a young clinician scientist who really wasn't quite sure where the job would be that I would come back to. My eldest daughter was just starting primary school, and the schools were a little bit different in the US as well, particularly in New York. And all of a sudden this opportunity came out of, to me, nowhere. And it was possibly the best opportunity I could have imagined to be able to practice clinically, but also to undertake the sort of research I was actually doing when I was in New York, but here in Australia. So it was an opportunity that I couldn't pass up.
Was there a relief from your family to be coming back, or were they suitably Americanized by then?
Oh no, they were very pleased to come back. I mean, my eldest daughter was just starting primary school, and I had 2 other daughters, and my eldest son was born there. So when you've got, a couple with 4 children in a 2-bedroom apartment in the Upper East Side in the middle of winter without a car, it's about time to think about when you want to come home.
Mind you, I don't classify New York as part of America really. It's New York.
It is very different.
Yeah.
It's true. It's true.
There's New York and then there's the United States. There's New York,
Yes.
there's California, and then there's the United States.
Yes, it is. It is.
So that, okay, exciting. So you've started your research really in New York.
Mm.
And you selected your pathway. So you would describe the pathway you selected. Why?
So I was fortunate when I came back to Australia that I was able to actually pursue the 2 main areas that from a research perspective I was principally interested in. The first was to use some of the really innovative new imaging techniques, particularly positron emission tomography or PET, to be able to identify where cancer was in the body. in ways that you couldn't do with any other technique, and monitor how treatments were impacting on the cancer. And on the other side, to start working on developing new treatment approaches, particularly with targeted antibodies, and to be able to not just undertake the basic research and development, but also go into human trials, and to start some of the very first human trials with antibody-based therapies in Australia. And that was because of my involvement with the Ludwig Institute for Cancer Research, and the fact they were developing a global clinical trial program that I became involved with. One of the interesting things that happened was that it was actually quite difficult to get antibodies made that would be of a suitable quality or quantity that you could use for human trials, or it could be very expensive. So the Ludwig Institute decided that it would actually build a small production facility so that antibodies or peptides for vaccine trials could actually be made for institute trials that were being conducted around the world. And that was actually decided to be built here at the Austin Hospital. And I was actually asked to take responsibility for that facility. And we did that. And we ran it for almost 10 years. And were able to produce antibodies and peptides for vaccine studies that were used in, I think, close to 25 sites in over 10 countries around the world, which was a fairly ambitious but very productive undertaking at the time.
Would you mind telling me, excuse me, why they're called antibodies?
So antibodies were first discovered and the name attributed to them by Kohler and Milstein, that were scientists that actually were able to create an ability to produce artificially made antibodies separate from those which are present within the normal body. I mean, antibodies were first identified, almost a century ago as being proteins which were probably going to be contributing in some way to the immune system. But we now know, of course, much more about what antibodies are, how they're made, what different forms exist in nature. But we've been able to engineer antibodies in many, many different ways now, which even 20 years ago we didn't realise that we could. And that's why they're one of the most successful new therapeutic approaches for cancer patients at the moment.
You might need to walk me through why or how.
Yes. So antibodies are part of the normal immune system in humans as well as in mammals and other animals as well. Their principal role is to recognise when something is foreign in the body. And so if recognised, such as a virus, the antibodies can actually mount a response, latch onto, in this case, a virus, and actually cause the virus to be killed. And there are various ways the antibodies can do that, through the actual effect of the antibody on, for example, a virus, or if it's an abnormal cell in the body, it can actually cause an activation of other factors in the blood which can actually cause inflammation and also death of the cell itself. So if the immune system is working properly, it's very effective at being able to remove abnormal cells. And so one of the reasons that cancers can develop is because the balance of the immune system in being able to control abnormal cells from normal cells can be disrupted. If the immune system works inappropriately, it can become too active, and that can actually result in autoimmune diseases, which can affect the skin or the bowel or many organs of the body as well. So it's a balance within the individual that the immune system tries to play an important role.
And every individual is different.
That's right. And so some of the more recent developments with antibodies is to create antibodies which can actually upregulate the way in which the immune system can recognise a cancer cell as foreign and therefore mount an immune response to those cancer cells and cause them to be attacked and actually die, and so therefore can remove the cancer cells from the body. What we've been more recently doing is trying to identify targets which are expressed in the cancers, either on the cancer cells or in the what we call the supporting tissue or microenvironment of the cancer. And then we can target those, and either by activating the immune system or by delivering payloads, drugs, toxins, or alpha and beta particles, you can actually home in and cause the destruction of these cancer cells specifically, while trying to avoid the toxicity of the normal tissues of the body.
So step one, target. Step two, take it out. Basically.
Yes. Yes,
And with as little collateral damage as possible.
correct. And of course, stimulating the immune system can be very effective. We've seen therapies approved, what we call immune checkpoint inhibitors. It can be remarkably effective. There are some patients that, 5, 6, 7, 8 years later appear to be disease-free. They may be cured. But some of these treatments can also have quite a lot of side effects, because if you stimulate the immune system, you can get a lot of quite obvious side effects which may require the treatment to be slowed down or even stopped for a while. So there's a balance that we have to observe even when we're getting very good therapeutic effects, to try and mitigate against the side effects which might occur.
So worst case scenario, if things go pear-shaped, it's worse than the disease itself, or?
I wouldn't say that it's worse than
In your own
yes.
words,
I would say that sometimes the side effects from the immune regulating antibodies can be severe enough that you have to stop the treatment and you can't restart. But in most patients, you can work your way through, you reduce the dose, or you have a pause in the treatment. There are various types of treatments that can be used for those side effects, and then you can restart the actual antibody-based therapies again.
Have you had any eureka moments in your life?
There have been over time quite a few times when I've seen a result from lab experiments, or been treating a patient with one of our new therapies and have observed something and thought, where did that come from? And it's that unpredictability that provides that frism of excitement. You think, this could actually be really important. Now, sometimes it is and sometimes it isn't. You can't predict when it's going to happen, but when it does, it's pretty special.
Do you have one in mind that you'd like to relate, or without naming names if you know what?
So there was an antibody that I was involved with developing, along with Dr. Lloyd Old, whom I worked with very closely in New York, where we felt that we had an antibody which could actually specifically target tumour, but the target that was expressed in normal tissues would not bind the antibody. And so I actually presented a lot of the data from our laboratory studies at conferences, and people were interested but not convinced. And so the very first clinical trial we did, we incorporated the ability to image where the antibody went in the body to prove if it would go just to the cancer and not to normal tissue. And that first patient actually revealed that it was true. And that was a very special moment as well.
So there was a home run, basically.
It was, and we completed the trial, and every patient subsequently showed exactly the same thing. And that was the study which actually led to us being able to go to big pharmaceutical companies and say, we think we've got something. And one of them ended up licensing it and has been taking it into very, very large trials to see if we can make it into a potential therapeutic.
Because that's the thing, research can only go so far.
Yes.
Excuse me. You get to the point, and then big pharma has to take over because excuse me, they've got the money. Would you like to express that in your own words? Because I'm coughing and I'm not doing this.
That's no problem. So, of course, making a discovery is wonderful, but to be able to take it into human trials is a major journey. It's quite complicated. It can take quite a bit of time, and it can take quite a lot of money. I've been fortunate to be able to involve with a number of journeys from discovery through into human trials. And there's no doubt that it requires not just the knowledge to be able to do that properly, but also the resources to be able to do it. And it's not cheap to be able to take a molecule into human trials. I think we're very fortunate in Australia that the biotech industry has really expanded over the last 5 to 10 years. So there are companies in Australia that are capable of doing some of the early phase clinical trials. But without doubt, if you really want to go into the large phase 3 trials, which lead to the...
Oh, I'm so sorry.
That's okay.
Is the water shoot?
I need something.
You got that?
Phase 2. I'm sorry.
I think we should probably cut, the car running out of car.